Hello, everyone, and welcome to another week's end. Welcome to another NeuroTools Webinar. We're very excited to have you all joining us today, and welcome to our YouTube viewers as well, once this gets posted online. Today, we're very excited to be hearing about the Human Connectome Project by Dr. Jennifer Elam. Dr. Elam is the scientific outreach lead for the Human Connectome Project and Connectome Coordination Facility based up in Washington University School of Medicine in St. Louis under HCP Co-PI Dr. David Van Essen. Dr. Elam organizes the annual HCP educational course, creates project documentation, and assists users in finding information, navigating, and using HCP data and software tools, including the Connectome Workbench software platform and the new BALSA database for sharing reference and user-submitted brain maps and data. The webinar today will go over the structure and the goals of the projects, how users get accustomed to access the data, where to find the information, and I believe she will be highlighting some of the advances already achieved and briefly demonstrate some of the tools being developed, including the Connectome Workbench software platform and the new BALSA database for sharing brain maps and data. Without further ado, I'd like to hand it over to Dr. Jennifer Elam, and we're very excited, and thank you, Dr. Elam, for taking the time to explain the Human Connectome Project to us.
Well, you guys are all very welcome, and I'm happy to be here representing the Human Connectome Project and its kind of follow-on projects that have kind of blossomed into this huge array of different kind of data sharing tools and analysis arms for neuroimaging data especially, but also behavioral data on humans. So I have slides, and then if we have time, I'm going to go into more of a little demonstration mode so that you can get your get an entry point into getting into some of the databases and tools that we share for our project. And you'll see when I can get to my next slide. There we go. Okay. So there's actually, there's quite a depth of information that I could cover. I'm trying to be kind of broad so that you can dig in deeper if you're interested in a particular aspect of what I'm talking about. Feel free to ask a question and I can point you in the right direction if you want more information on things. But I'm trying to kind of cover everything, and then we can dig in to a few things more fully. So first, I'm going to tell you what a connectome is, since all of you may not know that, and then kind of go into what our goals were for the original Human Connectome Project on young adults, how we collected, analyzed, and shared the data, which we're actually still, even on the first project that officially ended 2 years ago now, we're still actually doing further analysis and making that data available. And then we're going to talk a little bit about how you access the data. And there's various ways you can do that. And then tools that we've developed as part of the project and alongside the project to use to work with our data and your own data on the brain. And then we're going to talk about the impact so far. And then looking forward, we're talking about how the HCP has kind of developed a neuroimaging paradigm that we're hoping other projects do similar kinds of high quality data collection and data sharing. And then we also, around 2 to 3 years ago, the NIH decided, okay, the HCP Young Adult project went very well, so let's open it up to more projects that are the data is similarly collected and shared, that are on diseases that affect the brain, and also do a younger population and an older population of healthy individuals. And so that those projects kind of had a request for applications, and then we got also committed toward, since our database was so successful at sharing, we also were able to compete for a grant to become the Connectome Coordination Facility. So for these further grants that are doing projects on diseases and then on younger kids and older adults, we're going to be a centralized place for people to get their data or get information on all of these data sets and then compare them. So first, what is a connectome? So there are a lot of ways you can define what a connectome is. It's a comprehensive map of neuronal connections, and there's different levels that you can do research on neuronal connections. One is the macroconnectome, which is a whole brain, long distance connections between regions of the brain. And this is done with neuroimaging data. So that's mainly what I'm going to be talking about today. So in an MRI, the voxels, kind of the current technology is between 1 and 2 millimeters on a side voxels. Those are cubes of the brain. And you can obviously do this kind of imaging in vivo on, healthy and diseased individuals. They just have to, have certain things, like they can't have metal in their body or something like that. But otherwise, you're able to do this kind of imaging, and it's non-invasive, doesn't have any major health effects or anything like that. So that's nice for humans, but it is lower resolution than what some people are doing. You may have heard of Jeff Lichtman and his rainbow research, where they're actually imaging mouse brains slice by slice and building neuron to neuron connections and looking at synapses and all of that stuff. And you can get down to to micro scale precision. But of course, to do that, you have to cut up a little brain, and even on a mouse brain, that's a gargantuan task to assemble even a small part. So they've only really been able to assemble on the order of about the size of a voxel, or a few, of the mouse brain. Oh, okay. And then there's one in the middle that's called the mesoconnectome, and that's kind of cellular long distance connections, and that's where you're doing tracing types of studies. And you can do that in the mouse and in primates, but it is invasive, and it does require many injections, many brains. So for all of those reasons, these are all very important ways to look at the brain. But for the human brain, we really can't do the other two on any kind of scale. So we're going to focus on the macroconnectome. So we're trying to make that aspect of things the highest quality as possible with the Human Connectome Project. So as a history, in May 2009, there was a request for applications from the NIH Blueprint to do a large-scale project of around, I think they were, I don't think they were very exacting, but I think around 1,000 subjects is what they were hoping for, and kind of to push the edge of what we can do with neuroimaging. There was also kind of language in that RFA that talked about, using different modalities of imaging and perhaps doing genetics. So in 2010, September, 2 consortia got awards for this from this request for applications. One was our Washington University, University of Minnesota consortium. We got a large grant because we were going to scan 1,200 subjects, and we wrote in 2 years of methods development and piloting so that we could really push the edge of what we're doing. And then we wrote into the grant that we would actually build a new scanner at University of Minnesota, and then that would happen in that first 2 years, and then actually ship that scanner to Washington University in St. Louis and do all of our scanning on that one scanner at 3 Tesla, and then have some of our subjects also go to University of Minnesota for 7 Tesla scanning on their 7T scanner up there. And then about 100 subjects we would do at St. Louis University. They have a magnetoencephalography set up there. So we also do that kind of scanning, and then you could do cross-modal comparisons within the same subjects. So again, we targeted for 1,200 subjects, and we decided in order to leverage genetics to actually recruit twins and their non-twin siblings. So the way we did that was Missouri actually keeps track of all the twins born in the state and all the hospitals here. And so there's a study ongoing here at Washington University that's called the Missouri Family Study that actually interacts with these twins for their participation in research studies. So we were able to leverage that and recruit these people and their siblings. So we were shooting for an average of four siblings per family. I think we got somewhere between 3 and 4. It was probably on the closer end to 3, but that really helps with the power of our genetic data, which at 1,200 subjects is really kind of on the edge of when you can do any kind of really useful genetic comparisons between people, comparing them to their behavioral data and their neuroimaging data. So again, we had different kind of parts of the project that were different people were specialized in, and some of our group is actually at University of Oxford in England. They're a group that develops neuroimaging software tools called FSL is the suite of programs. So they were involved. And then we also had some people on the magnetoencephalography MEG team from Italy, Germany, and the Netherlands, because they happen to be particularly good at analysis of that kind of data. The other consortium that also got funded was at Massachusetts General Hospital and UCLA. They actually moved to USC subsequently, and they developed a very high-end scanner particularly for diffusion imaging. So that was a 2-year study, and actually when you Google Human Connectome Project, you end up getting their site actually. So that's just something to know. When you see those really colorful images of brain tractography that are kind of streamlines, the band Muse actually put one of those streamlined pictures on their one of their album covers. Those are actually from that other project. We have connections and we're colleagues with them, but we're not the ones that produced all that stuff. So just so you know that. And then in 2013, while we were in the middle of our main HCP project, the NIH came to us and said, would you be interested in applying for this pilot study to study young kids and older adults using the same types of methods that you're doing in the HCP. And so you don't turn away a request from the NIH. So we prepared a grant and was awarded a supplement to do that study. And that was really interesting experience, because we ended up having to adapt things quite a bit to those two age groups, even with normal subjects. Young kids tend to wiggle a lot in the scanner, and they can't do all the tasks that the adults do. And the older adults also don't like being in the scanner, won't tolerate being in the scanner as long and stuff like that. So we had to adapt things for that, and that also included we on the main HCP, and I don't know if I discussed this anywhere else, so I'll bring it up here. On the main HCP, the scanner that we kind of custom developed with the help of Siemens ended up not being what Siemens decided to market as their main product going forward for people who are looking for a 3 Tesla scanner. So one of the things that we did in the lifespan pilot was also to compare kind of the next generation scanner, which is called a Prisma, that was to be kind of available widely. And so we were able to kind of make sure that the data we collected on our customized scanner and the Prisma scanner could be compared equally. So then in 2016, and I guess I left out a time point here, in between there, the lifespan pilot was successful enough that the NIH decided to send out another request for applications for lifespan projects to do full-scale 100 or 1,000 plus subjects at the younger ages, and again 1,000 plus subjects at the older ages. And we actually, our consortium plus several other people that are particularly good at working with those age groups at multiple universities, because to get the numbers that we needed, we can't all do the scanning here at Washington University this time. We were awarded 2 of these grants, one for the development, which is 5-year-olds to 18-year-olds or 21-year-olds, and the HCP Aging 36-year-olds up to 90-year-olds. And then another group mainly based at University of North Carolina got this HCP Baby grant, which is for newborns to 5-year-olds. So our goals for the main HCP project was to study 1,200 healthy adults. Like I said, there were twins and non-twin siblings, and we were using cutting-edge neuroimaging methods at both at 3T and at 7T, advanced pulse sequences, which means we were doing multi-band parallel imaging. And what that means is that you can basically maximize the amount of data that you're collecting per unit of time. So we were actually getting like 70 images for every TR that we were collecting. So that actually made the data much richer, as far as, you're able to do better statistics, get more information from each second of data that you have. And then we were collecting multimodal data, structural MRI, diffusion MRI, resting state MRI, and 7 different tasks. So this amounts to 4 hours of imaging. And then for the MEG subjects, they spent a couple of hours over at St. Louis University getting that kind of data, both resting state and 3 of the tasks. Then we also did, on every subject, we did behavioral testing and took blood and saliva samples for eventual genotyping. And then the major goal was to share all that data in a user-friendly informatics platform. So when we went to acquire the data, we used these improved scanners and pulse sequences. Again, we got this multimodal imaging. Our data quality was exceptionally high. We actually did extensive quality control on every subject. And we were able to reach our goal of 1,200 subjects. We have, I think, 1,206 that made it through. We had a few subjects, you know, have to leave the study for various reasons, but we have 1,206 that have behavioral data in our database. I think it's 1,113 subjects that have some MRI data collected. So some subjects just, you know, for various reasons, again, claustrophobia or whatever, you couldn't get good MRI data from them. So that's the difference in numbers there. But really we were really proud to get that many subjects with MRI data. And we actually have many subjects that completed everything 100%. It's like over 900 subjects that did absolutely everything. So that's really amazing. And a lot of twin pairs that we were able to get 100% on. Then we had 184 subjects scanned at 7T. Again, they had to fly up to University of Minnesota for that. And then the behavioral data we collected was 478 different subject measures, and that's anything from demographics to cognitive scores on assessments. And I think we've got a slide in here that kind of goes more into detail on that. And then for the MEG, we had 95 subjects complete that. And we did, like I said, task and resting state for that, and then blood for genotyping. The genotyping work has been done, and we've sent it to dbGaP, but we're working through kind of the release process on that. So that should be out pretty soon. There's a lot of people waiting for that data. So just to kind of go into a little more detail on a couple of the different modalities. For structurals, we did T1 and T2 weighted images. And if you're familiar with neuroimaging data, you can look at this example image and see how clear this structural image ended up being here. And then we ran all of the data for every subject through FreeSurfer and got segmentations and surfaces through that. And we also did QC on those to make sure that there weren't any weird abnormalities from running FreeSurfer. If we did see that, then we'd run it through again, and sometimes that would just fix the problem. And other times, there would be an anatomical problem with the subject that would be revealed through that process. And those subjects that have those kinds of issues, we actually have a flag in our database so that you can know who those subjects are and take them out of your analysis if that will create problems for you. So another thing that was really good about collecting both T1 and T2 weighted images is that we could do myelin maps, which are down here on the right in the color. Those have been really valuable for doing proper brain registration between subjects and getting better group average data through those better registrations. Okay, and then at 3T, we did 7 different tasks. And part of the reason we did all these different tasks was to make sure that we were catching all the different kinds of brain domains that that people might be interested in, and to also be able to use that data that kind of lit up different parts of the brain, so that in aggregate, when we look at this down below on the left, in aggregate, we're actually getting nice, good coverage of brain regions through what gets activated by these tasks. that actually helps us in registering individual brains, again, to each other so that we get, more in register, so that we know that when somebody has a resting state network that has, a few different regions lit up, we know that how that compares to another person when they have their networks lit up. So that's really an important part of kind of moving the whole neuroimaging field forward. And then at 70, we did some different tests. We did retinotopy, so that's for visual cortex, and then we also did these natural movies that had that a group, the Gallant Lab, actually helped us to kind of parse out different parts of those movies, so that we know what's going on when you can actually time block what's happening in the brain to when the subject is looking at a certain part of a video.
Hey, Dr. Elam?
Yes.
You do have a question from Dr. Bandrowski in the chat. If you'd like, I can read it to you now.
Yeah, please.
What is the coordination like between the HCP and the ABCD project that is now heading our way? The subjects are certainly healthy children, so I should be able to look across these data.
Right. So there's actually been a lot of coordination between the development of what we're doing in HCP development with our kids and what they're doing with ABCD. Part of that coordination is that Deanna Barch is actually on both projects, and she is the head PI actually for our HCP development. So we're using similar protocols for the scanning at MRI, and I think there's also some overlap in some of the behavioral tests that we're doing between the projects. We've definitely got an eye on people wanting to use both data sets together. So we're going to make the database helpful for that kind of comparison and make sure that the information is out there on what things are the same and what things are different between those two projects. Hopefully that answered you. I mean, it's we're kind of just now to the point where we're going to start getting some of that information out even before data is available for either projects. But we're probably a couple months from getting that kind of really clear on the website.
And Anita says, Dr. Bandrowski says, thank you from the chat.
Oh, you're welcome. Okay. So what kind of behavioral tests did we do for HCP Young Adult? So Deanna, actually, I should have gone to the slide to show her picture a little earlier while I was answering. She kind of led the effort on this aspect of the HCP because she's a psychologist and is kind of aware of, what has become the canonical types of assessments for various neuropsychological, things that you want to know. So another thing that we were that was part of the RFA was to use the NIH toolbox, which had been being developed kind of right before HCP started. And we actually were the first major project that used NIH toolbox kind of broadly, which was an interesting thing because we were coming up with names of, like some of the data we actually named some of the data points for Toolbox because we were the first ones using it widely. We also use the PIN neuropsychological battery, which is nice because it's a computerized test that subjects can do. And then it automatically, so it's web-based, so they get the data immediately, and then they send us back the analyzed data. So these were collected on diverse phenotypes, and some of the cognitive things were like match the shape in that one, and some sensory tests, personality tests, and then we collected a lot of psychiatric sorts of measures and substance use measures. Because some of this data is sensitive, whether or not you used various kinds of drugs, and whether you have a family history of psychiatric disorders and so on. Plus there's PHI, BMI, and thyroid hormone levels and that kind of thing. Some of that data is what we've put under this restricted access, and that actually requires a special application where you and if you are not a PI, your PI as well has to sign up at the same time to get access to that kind of data, so that people aren't allowed to, or people aren't able to find out who the subject is by looking at all of the data that's available, without some check on the system that you're an investigator, not going to try and find out who that person is. It's especially important with this study because it was twins and their siblings, so a lot of families knew that their sibling was part of the study, and so they might want to try and find out what their sibling scored on some various tests, or whether their sibling had used drugs before or something like that. So so this kind of allows us, having that application allows us to weed out anybody who's not kind of a certified research person who's agreed to not try and figure out who people are. So then we, the way we analyze the data, we made a lot of improvements to the what we call the pre-processing pipeline. So they use a combination of FreeSurfer, FSL, and Connectome Workbench, specifically Workbench Command. Those are the three programs that you need to have to run the pipelines. These pipelines are basically scripts to kick off all of these different processes that you do on the data, and that's all available on GitHub and documented there. We also have, I think I mentioned at the very end, we have a HCP users listserv email list that people ask questions all the time about how do I get the pipelines to work on my system, and so on. We also have made better visualization tools through the Connectome Workbench View platform, which I'll show you. And then we have done advanced analyses that we've made available to people, and these are some of them. I'm not going to read through these, but these are some of the data that we've made available. And we share the data. So we have the Connectome Database. ConnectonDB is what we call it for short, and it's a robust infrastructure based on what's called XNAT. So that's a databasing software for neuroimaging studies. We've had multiple data releases. I think we've had 6 major data releases. The latest one was the 1,200 subjects release in March of this year. And then we've released our 7T data. We released preliminarily last year in June, and then we released the rest of the subjects along with the 1,200 subjects release. And then the MEG release happened, the final MEG release actually happened a couple of years ago on the 95 subjects. We released extensively analyzed data. I kind of talked about that. And then the BALSA database, which I'll talk about in a little bit, is also a way to put out highly analyzed data in a format that's easy to kind of download and load up in Connectome Workbench and then get going on. So the kind of nexus for finding everything is the humanconnectome.org website, and here's what it looks like when you go there. And we've actually made a, this summer we switched over to a new website. It used to be only about the HCP Young Adult project, but now since the project has grown into more projects and we've also gotten the grant to be the Connectome Coordination Facility, this is now a single hub for all of those places, and then you can click on various types of projects or types of things you want to learn about or get access to kind of navigate your way through those different parts of this major now kind of ecosystem of projects. So how are we doing on time? Okay. So I mentioned restricted access. There's also just open access that you only have to agree to a few different stipulations, and you can do it electronically to get access to the data. You can access the data via ConnectonDB. That's our main access point. There you can download data, you can filter subjects, you can just look at the data, look at the behavioral data in there, or you can download a CSV that has those measures in it. You can also order what's called Connectome in a box. That's hard drives that we load up with data and ship to you. So that's kind of a way to get a large amount of the data. This is very large and it takes a long time to download. We've also made our data available on Amazon Web Services. So again, our visualization has the Connectome Workbench platform, and you can make cross-modal, cross-subject comparisons there for discovery. So what is ConnectonDB? It's this database that has kind of a front page, and then you can get into a subject dashboard where you can look at a whole list of subjects and look at their data measures on various things. So it's a way you can get, you can kind of navigate that data, but also download imaging data. So we make available both unprocessed data, so it's completely, the only thing that's happened is that the DICOMs off the scanner have been converted to a smaller format called NIFTY that you can use, and then minimally preprocessed data, and then more analyzed data is also available there. You can do different searches, and there's some group average data there, but then we also saw that there was a need for user submitted. We had got a lot of requests for people to submit their data to a database so that other people could look at their data for their subjects and their studies. So for that, we actually have another database that I'll tell you about in a minute called BALSA. So this is the page you come to when you go to db.humanconnectome.org, and you create an account, and then you can log in and you can see those pages that I show you here. So I just had to register, and then I want to tell you about Connectome Workbench. So this is kind of you can download it from the humanconnectome.org website. It's under the software tab, and it's a standalone software platform that comes with both Workbench View and Workbench Command. So Workbench View is what you see here, this GUI that allows you to load up data on brain structures and look at it, and you can also draw on it, you can annotate, you can basically make a figure within Workbench if you want to, and then you can save your whole workspace as a scene which is really, really helpful, because then you can, you know, give somebody your scene and all the files that are in that scene, and then they have everything they need to be able to kind of look at exactly what you were looking at and make their own judgments on, you know, on what your analysis was like and everything like that. So that's a really nice feature about Connectome Workbench that really dovetails into the BALSA database that I'm about to talk about. So Workbench Command is a command line tool that has a lot of different kind of utilities for doing different things on imaging data, both the data coming straight out of the scanner and the pipelines, but also data that's been kind of put into a different format for looking at things like connectivity between different regions and tractography and that kind of thing. So here we come to the BALSA database, and this is the Brain Analysis Library of Spatial Maps and Atlases. And this is a fairly new addition, but again, this is really designed to be a place where people will create a data set of their own data, usually attached to a publication, and then upload that to our database so that people can then look at it, look at thumbnails and that kind of thing, but then they can also download that data and really interact with it. And right now, I mean, we have plans to make this kind of accessible to multiple visualization platforms, but right now it is Connectome Workbench specific, where you make a scene in Connectome Workbench, you upload it to BALSA, and then you can share your data that way. So that's where it is, balsa.wistol.edu. And I think this is going to say what I just said. Yeah, you upload scene files. And you can also publish your link to your submission in BALSA. So that kind of makes things easy for getting to the database and getting to the exact data set that you want. So what is our impact for HCP Young Adult? We've had over 9,000 investigators accept the HCP data use terms, and almost 1,000 have agreed to the restricted access data use terms. Over 14 petabytes of data has been shared, and that's a lot of data. So 9.4 petabytes has been downloaded, which is kind of amazing. And 5 petabytes has actually been requested shipped on hard drives. So we only charge people the cost of the drives, but that's a lot of hard drives too that we've shipped out. Again, the data is also shared on the cloud via Amazon Web Services. We actually don't know how many people use the data that way. We haven't been able to get those metrics, but we've heard a lot of people using it. So I think that's also getting a lot of traction. And this is a wiki page that allows you to find out how to connect to it that way. There have been over 430 publications using HCP data, and over 350 of those publications are from authors completely outside of the HCP consortium. There's 100 people in our HCP consortium, so that actually is, so there's actually a lot of people that I'm excluding, you know, from that list there. So it's actually here's the whole group, and we had 10 institutions involved. So it was a big project. It looks like we're getting kind of short on time, so maybe let me just skip through a couple things. We published this paper, the HCP style neuroimaging paradigm, to kind of give the community kind of our collective wisdom from going through this HCP project experience. And we came up with 7 core tenets, and pretty much they come from what I've already said, where we're collecting we recommend collecting lots of data, maximizing your resolution, minimizing your distortion, respecting the geometry and anatomy of brain structures, aligning the data precisely, analyzing the results using an accurate brain parcellation, and freely sharing the extensively processed data. So I think that was really a contribution to the field, at least kind of put into one paper, kind of our collective wisdom on the project. So now we're going into this next generation of projects, which we talk about the project that I've just been talking about here on the left from 2010 to 2016, and then going into the next phase where we're doing lifespan HCP. There were 14 projects that were funded in the Connectomes Related to Human Disease project RFA. And then there's also this project in the UK called the Developing HCP that's actually prenatal imaging. So they're doing from 20 weeks post-conception through, I think it's 2 weeks after birth. So that's a really interesting project that they've had their first data release. And we're actually going to share some of their data on our Connectome Coordination Facility, ConnectonDB site. And then as we alluded to before, the ABCD project is going on, this major 10-year study of the adolescent brain. And we're going to share their protocols so that you can make direct comparisons between our protocols and theirs. Their data is going to be hosted in a different platform than CCF. Okay. So the CCF is just on kind of a broad level. It's committed to being a help desk for people who are trying to acquire their data in the same sorts of ways, the HCP style way. And then we're maintaining and expanding HCP informatics infrastructure. That means all of the tools that I've talked about are going to continue being developed. And then we're going to serve as a harmonization center to try and make these differing projects as comparable as possible. So here's the whole list of all these different projects that are going to fall under CCF. The first 4 are the lifespan projects, and then the next 14 are the other Connectomes and human disease projects that are being conducted by different research groups at different universities than here. So you can look through that and see what you might be interested in. So just to kind of touch upon the lifespan child development that we're doing here, it's 1,500 subjects, 5 to 21 years old, and we actually have a couple of different aspects of the project. There's a cross-sectional part and a longitudinal part that will span the puberty years. So that'll be very interesting to get 2 different time points from the same subject. We're trying to do recruitment based on kind of the breadth of the human or the American population. So we're trying to get hit targets of different age groups and different ethnicities and SES levels so that people can do those types of comparisons. We've got a protocol that we modified for those different ages of kids. The older kids, the ones in the late teens, they're doing, I think it's from 14 and up, they're doing basically the same kind of protocol as the adults in the lifespan aging group. But then the younger kids, they can't do as long in the scanner, so they've got a slightly different protocol that we share. And then for aging, we're looking at 1,200 subjects. And some of those subjects are also longitudinal around the perimenopausal timeline. But I think we're doing both men and women along that, in that age range. So, so there's a lot more data to come, and keep your eye out for that if you're interested. Like I said before, we have this HCP users listserv that's really active. We have daily emails on the list, and that allows users to really interact with our scientists and get answers to, you know, how do we do that? How do you make your data work with our pipelines and so on. We also have this HCP announce list that announces data releases and things like that. So even if you're not interested in knowing the kind of nitty-gritty details on how you handle data, this HCP announce list is nice because you can hear about major happenings for HCP.
Excuse me, Dr. Elam.
Yeah.
Is there a URL I could type into the chat box for that last slide, just so people could click on it and just sign up right away right now if they'd like to?
Okay, so it's actually, let me just get out of this, and it's
Sure.
humanconnectome.org, and then if you go to the contact, let's see, if you go to contact us, so it's /contact us, humanconnectome.org/contact us.
All right, sounds good.
In between.
You might hear me typing for a second. I apologize. /contact us with a hyphen in between.
Uh-huh.
Bummer. Looks like I'm getting a 404 error.
Oh, did you use www?
Yes. .humanconnectomeproject.org.
Nope. No project. Just .humanconnectome.org.
All right, you got it. .humanconnectome.org. Here we are. Got it. All right, I will paste this into the chat. Thank you very much. Sorry to interrupt.
No, it's okay. Does anybody else have any questions? I know we're running short on time, but anybody?
I have a question from Dr. Bandrowski. SFN ?
I wish we were still funded to do things like attend SFN and share, have a booth and talk to people about our project. But unfortunately, money is short these days with the lifespan projects being at 4 different sites. So we've got 4 different groups of research assistants running people through scanners and all that kind of stuff. So, so I don't get to go. Sorry.
Well, that is unfortunate to hear. Marianne also has Dr. Martone also has a question. What do you think the useful lifespan of the data are before the technology improves so dramatically they have to be taken again? ?
That's a good question. I mean, it's, I think probably we're looking at 5 to 10 years of lifetime. I mean, this is what I've heard from others who've tried to conjecture on that, but it's also hard to know exactly what's going to happen technology-wise. We really involved people that were kind of the drivers of a lot of the technology advancement. So Camille Ugerbil and Isa Yakub at University of Minnesota are really at the forefront there, and they haven't said, oh, we really should have done this because we're limiting the lifespan of our data, but it's also hard to know. I mean, there might be, with the multi-band imaging technology, we were really lucky that happened right before we started doing our piloting, which really allowed us to take advantage of it. And we don't know if another another advance like that might happen. And if that did, then I'm sure people will be collecting data in a different way. So.
And Dr. Bandrowski would also like to follow up. Is there any way we can help spread the word at SFN at our Neuroscience Information Framework booth?
I could probably put something together that you guys could have sitting out there to hand out.
That'd be wonderful.
That would be really nice for us too. Thank you.
Absolutely.
So that's coming up though, right? Like in a few.
That's correct. Yeah, I believe not too long at all.
Okay.
The end of next week.
Yeah, so I'll get on that then.
All right. Excellent. Thank you. Well, it looks like the questions in the chat are starting to wrap up. I'm getting a thank you from Dr. Bandrowski, and thank you from myself personally, and thank you from Dr. Martone as well. And is there anything else you'd like to add, Dr. Elam?
I guess the only other thing might be if you're on our website and you go to well, actually you have to go to the main part of the site and Young Adult HCP, go to that project, and then you have to click on this here to get to the page that's specific to the project. There's this tab here called documentation, which is what a lot of my effort goes into the maintaining the documentation for the projects. And if you click on any of the data releases, you end up getting this whole page of documentation that's available. You get this huge PDF that is over 200 pages long, and it basically touches upon the entire aspects of the project. We're going to be producing this for the lifespan projects coming up too, but this is really a really good resource and has lots of links and stuff in it to kind of navigate where we put all the different data sets and resources that you might need. So that's a good other place to really get to know the project.
Great.
Okay, well, thanks everybody.
Thank you so much for doing this webinar. And I just want to add that in a few hours, we will have this webinar online. I'm going to do some brief edits just at the beginning of it so that it starts right when you started presenting. And then we'll be putting it on our YouTube channel, which is accessible through the New Info Upcoming Webinars tab. If you just go to Dr. Elam's section on our webinars, you will see a YouTube link very shortly. I just want to say thank you to Dr. Elam for taking the time to do this webinar. The next webinar, Dr. Pollock asks, is next week at the same time. It'll be the Mutant Mouse Resource and Research Centers, MMRRC, National Consortium, presented by Dr. Kent Lloyd. That'll be on November 10th, which is next Friday, and it'll be at 11 a.m. Pacific Standard Time. That's also accessible through the link that I provided in the chat box, newinfo.org/about/upcoming webinars. Thank you again to everyone who joined us. And like I said, this webinar will be online momentarily in a few hours. Okay, I'm going to end the webinar now. Thank you very much, Dr. Elam.
Thanks.